Good evening, everybody. What a great turnout on a rainy evening. We've got our traffic director here. What a special evening. Thank you for coming. I'm Randy Heiler. I'm the co-chair of the Spotlight on Science Committee. And before we start, I'd just like to remind everybody, including myself, to turn the cell phone off. Make sure that it's silenced. Yes, it better be off. Thank you. And we'll have some opportunity for questions at the end as well, so please hold them for later. On our behalf, I'd like to welcome you all to our second presentation of what is now our second season of the Spotlight on Science program. And judging by the attendance tonight, it looks like we've been pretty successful in adding a new program to the Newport Beach Public Library already outstanding roster of distinctive programming, which brings us together to hear from the world's leading authors, artists, journalists, doctors, scientists, and so forth on topics of interest, relevance, and importance to our community. The objective of the Spotlight on Science series is to pull back the curtain on scientific research in an understandable and accessible way, and to highlight the importance and potential impact of these endeavors on our lives in our communities. We will be covering topics ranging from the infinitesimal to the infinite, exploring the sciences of biology, chemistry, physics, optics, neuroscience, electronics, the environment, and cosmology. Our 5 broad topical categories include the human body, our planet, energy and transportation, the worlds beyond Earth, and emerging technologies. Earlier this season, we were fortunate to hear from Dr. Jack Brower on the important role hydrogen will play in California's clean energy future. And last season, we focused on artificial intelligence, both as a breakthrough technology in itself and its impact on scientific research in tracking glaciers, water, coastal erosion, and flooding, all topics directly impacting our community here. And hopefully, some of you were able to attend the inaugural event on May of 2023, when we had the first talk covering the mysteries of the universe as seen from the James Webb Space Telescope. And I'd just like to remind you that all of these programs are available in our archives online at the Newport Beach Public Library Foundation website, nbplf.foundation. And also please calendar our finale of this season, which will be Dr. Lucy Jones, who will come to discuss California's tectonic activity, which has been coming along here in the last few weeks, and understanding the risk of earthquakes on Wednesday, April 30th. I'm apologizing for my cold recovery here. Hopefully most of you are also familiar with the Library Foundation's other major programs. The Witty Lecture Series showcases distinguished speakers. The Library Live brings authors of renown to the library. The Financial Literacy Workshops provide up-to-date information on managing your money and your estate. And Medicine in Our Backyard highlights the latest in health and medical advances. Plus, we have the Book Discussion Group, which offers a lively conversation on books of great interest and acclaim. But the Foundation also supports innovative library resources like the Media Lab, self-checkout computers, the Tech Toys, the Wayfinder, and the eBranch of digital resources. Because we cannot present these many programs or fund important resources for the library without your support, we urge you to join or renew your membership, which benefits all Newport Beach families and our communities. I'd like to note that I heard at one of our foundation board meetings the other day that we have 3 million patron visits per year in the library series here. That includes our online and in-house. So yes, it's a fantastic resource for our community, and I want to make sure everybody is aware of that. Especially because, as you can see, this is a room that often fills to and past capacity. It has limited occupancy and sight lines and audio, including the speaker. So what we have embarked upon is accepting a challenge to raise 50% of the cost of a new state-of-the-art library lecture hall, which is really going to be a community civic center, and it's going to be for the entire community. It's now called Whitty Hall. You see it going up in the parking lot. In recognition of a generous founding gift by Bill Whitty and Keiko Sakamoto, this will truly be a community-wide asset and will support literary, cultural, artistic, educational, and even business events for many years to come. So the construction project, as you can see, is now underway. and we hope to open the hall in about a year from now. But commencing construction does not mean we're finished raising the money. Our portion of the bill is about $12 million. The good news is we've raised about $11 million of that so far, but we have to finish. Thank you. But we still have $1 million, and it better be in the bank by December or we're in big trouble. So there is a donation can on the way out. We do urge you to help us make this vision a reality. And I'd like to point out that this library itself was also a public-private partnership done 30 years ago, right? 31. Here, and this is the reason we have this facility. It's this kind of engagement of the community and the city that makes this happen. It will just add so much to our programs, and we're going to be adding new big screen video technology and state-of-the-art audio-visual to the new theater, and we'll also have another 100 seats to host these events. Since the can may overfill with your donations, I'd like to introduce you to Jerry Capel, our CEO, who's happy to take your credit card or your check, any other form of donation, and our website is also obviously available. So there's the preamble. I'd like to introduce our topic and our speaker tonight. Tonight we're very happy to have Dr. Grant MacGregor, who serves as professor of developmental and cell biology and as the scientific director of the transgenic mouse facility in the Charlie Dunlop School of Biological Sciences here at UCI. He's also a member of the Chow Family Cancer Center. He received his bachelor's degree in molecular mutagenesis and DNA repair, say that quickly 3 times, from Sussex University in the UK. But it was his postdoctoral research where he started focusing on human genetics, gene therapy, and developmental biology at Baylor College of Medicine in Houston, after which he established his own research facility at Emory University School of Medicine in Atlanta. Then in 2002, to our great benefit, he relocated his laboratory here to UCI, where his research center is focused on understanding causes of human genetic diseases and age-related conditions using strains of genetically engineered laboratory mice, examples of what you'll hear more about tonight. Dr. MacGregor has also been notably recognized for his excellence in teaching during his tenure at UCI, receiving the Dr. DeGallo Professor of the Year Award, the Dean's Honoree for Teaching Excellence, the Golden Apple Award for Excellence in Teaching, and the UCI Chancellor's Award for Excellence in Fostering Undergraduate Research. And for those of us who had the benefit to spend some time with him before the speech tonight, it's obvious why he gets these accolades. I think we're all going to learn a lot. I think we're all going to enjoy the presentation. I'd like to welcome Dr. MacGregor to the library.
I wander around. You need to excuse me for that. Can everyone hear me okay at the back? You fine? Great. So it's a great honor to be here tonight to try and share some of the knowledge that we as the scientific community have acquired and the technology and the methodology that is now being used. In particular, what I'm going to focus on here is CRISPR-based genome editing. Many of you may have come across this in the news and online, so I'd like to sort of try and demystify things for you and give you a realistic appreciation of what is possible currently with CRISPR and hopefully what will be possible in the years to come. So I'm going to tell you a little bit about making woolly mice. I don't know if this is in the press yet, but it will be shortly if not. So these are mice that look a little bit like a coat from a woolly mammoth. Okay, this is a sort of party piece that this company in Dallas has done, but the company, as you'll see later on, is more serious about trying to revive extinct species, and this has major implications for conservation programs around the world. I'll tell you a little bit about how CRISPR has been used to make rice healthier. If you're like me and your physician looks at you with a scowling eye and says, you know, your A1C is a bit too high, and I go, oh, I don't get out too much. Well, you know, brown rice, things like that, to reduce the glycemic index, or basically lower amounts of sugar, can be very healthy. So for all the diabetics in the world now, the wide availability of CRISPR-generated rice that has a low glycemic index is something that's becoming popular. And then I'm going to tell you a very nice story that started back in 2019. And this was a company that Jennifer Doudna and Emmanuel Charpentier, two investigators who co-received the Nobel Prize recently for development of CRISPR technology and its application in medical realms. And this is now being used to cure sickle cell disease as well as a variety of other genetic diseases, many of which, unfortunately, some of you may be familiar with family members or friends that are affected. Okay, so I'm going to introduce you also to these little molecules up here, and I'm going to take some time and we'll go through this slowly and make sure you all have a firm foundation. Now, I don't know, I should ask Randy, do you all prefer to wait until the end to ask questions, or would you like to ask something in between? Yeah? At the end? Okay, we'll keep it to the end. If someone has a burning question, please feel free just to raise your hand, say question, and we'll try and stop, and it's probably because I haven't clarified or need to clarify something. So I'll do my best to try and do that for you. Okay, so without further ado. So here's a sort of smorgasbord. Here's our menu for this evening. I'm going to take you back to school to start with, and we're going to talk about the genome and how DNA makes RNA, makes protein, which is super important. Without that process, none of us would be alive right now. I'll then tell you a little bit about the discovery of CRISPR. This came from purely basic research. No one was looking for the CRISPR gene or the CRISPR locus or how CRISPR worked. This is something that scientists stumbled on roughly 40, almost 35 to 40 years ago now. And it only came over a slow period of time. Once again, emphasizing the importance of basic research to our advances. Scientific research is a cycle. We have advances in technology that produces new discoveries, and that allows development of new ideas and hypotheses. And that circle goes around and around and around. So new technology development is incredibly important to the engine of scientific discovery. I'll then give you a very brief review of some of the examples of how CRISPR is being used, including development of animals with modified traits and development of crops that have modified traits, including rice that we just mentioned. And then I'll give you a brief overview of using CRISPR to treat human genetic disease, because that probably is where most of you have a great interest. I would suggest to you that this is already underway. The FDA has approved a few treatments for this, but over the next 10 to 15 years will be, I predict, an explosion in effective therapies that can combat things that were once upon a time deemed extremely debilitating. And we'll cover some of those diseases as well. And then at the end, perhaps as a segue into a discussion, I'll touch on very briefly what we've done in the past, we being the scientific community, and where we are now, and then what some of the considerations are for the future use of CRISPR, including balancing pros and cons and concerns that people may have. Okay? So, let's start off with going back to school. Here is a cartoon showing a cell of some sort in our body. It's a rather sort of amorphous cell. The important thing here, we have billions of these in our bodies. The important thing here is in the nucleus is where the DNA is contained. All of us here, hopefully, have inherited 23 chromosomes from our mom and 23 from our dad to give a total number of 46 chromosomes. And these chromosomes, if you stretch them out, you would unpack them and form what Crick and Watson, about 70 years ago now, discovered along with Rosalind Franklin as showing the DNA double helix. Okay? If we schematically unwind the double helix, you can see there are a series of letters here, A, G's, C's, and T's, and what these denote are bases. So these are chemical bases which have the property of being able to pair with each other through some relatively weak bonds, but which collectively form very strong linkages. So Velcro, for example, if you take Velcro, one little hook and one loop, there's not much there, yeah? But if you take two pieces of Velcro with a lot of bonds such as this, then it's quite strong. So a wacky analogy, as I like to tell my students, but hopefully you get the idea that DNA is quite a stable molecule because these bases are held together through these special type of bonds. So this molecule, as you'll see, is an information molecule containing all the information for development and for life and for reproduction of organisms, including our cells. Okay, so here are a couple of individuals with their chromosomal content. If you were to look inside the genome of all the chromosomes, you would come across regions of letters, and you wouldn't be able to identify what those letters were telling you. But from many years of research, we know that collections of letters can form what are called genes. And genes, basically in the DNA, have a part that tells the cell when to confer an activity that leads to copying of the DNA to make an intermediate information molecule called RNA. DNA is double-stranded, RNA is single-stranded, and then this RNA is processed to remove these so-called intronic sequences, and that leaves behind a messenger RNA. And this contains the information necessary to form a protein. And proteins are made up of individual amino acids all bolted together. Okay? That protein then will fold in a particular shape and fulfill its function in the cell. It might be a structural protein to make maybe the nuclear envelope, or it might be a protein such as an enzyme to allow you to digest sugars, or all sorts of things. So here's a very simple overview then. Here's our double-stranded DNA with our 4 letters. Here's a messenger RNA that is being transcribed and formed from the DNA template. And then the messenger RNA can then be translated. And here are an example of a few amino acids that have been encoded by these triplet bases. Okay? So this is called transcription, this is called translation, and it forms polypeptides, which then make up proteins. The only difference really between DNA and RNA is that DNA is double-stranded, RNA is single-stranded, and DNA has a T in it, whereas that is replaced in RNA by a U. Okay, all good so far? Excellent. There will be a test at the end. Okay. All right, now here's where things can go wrong. DNA replication is extremely accurate. It has very, very high fidelity, but even it sometimes makes mistakes. When we go out in the sunshine, the DNA in the cells in our skin can get damaged as well. And the chemical damage can lead to changes in the bases. So sometimes in a coding sequence, for example, the DNA may encode for a series of amino acids. This is a serine, a valine, a proline, and a tyrosine. But if the DNA gets damaged and the base gets changed, so this C is changed to an A here, all of a sudden, in this case, the amino acid is altered. Instead of a proline, we now have a threonine. Sometimes those changes can be innocuous, and they actually contribute to the diversity that you see. If you look at our hair, if you look around the room, you'll see different types of hair. Some may be frizzy, some very straight, some curled, etc. Those can be due to what we call polymorphisms, or innocuous changes in the amino acid sequence in some of our genes. But in other cases, these changes can be deleterious. So in some cases, you may end up with a protein that no longer functions. This is depicted here. Here's a protein that has this spiky part here. It looks like my hair back in the 1980s. Okay? This denotes a part of the protein that is functioning. When you change it, the depiction in the cartoon here now says this protein is no longer functional. That may be tolerated by a cell. But in cases where the shape of the protein changes, it may affect the cell in a deleterious manner. So this is a classic example of a family of disorders called amyloidoses. And there are myriad neurodegenerative disorders and other disorders of other organ systems due to proteins that misfold. Prions are one of the key classes of this, discovered many years ago now. Okay, so The bottom line here then is the ability to selectively edit DNA and to change DNA sequences is a very powerful tool that could allow development of novel therapies for disorders, perhaps due to changes in DNA sequence. And they could also be used for myriad other purposes, for example, in biotechnology, and some of those we'll talk about in a minute or two. Okay, all good so far? Excellent, well done. All right, so now we're going to, big part, we'll go slowly here. How was CRISPR discovered? Okay. All right, so here's a bacterium, a lowly bacterium. They're not so lowly, they're super important to our health. The bacteria in our guts are really, really important. They help stimulate our gut, for example, to produce something called serotonin, which is an important neurotransmitter that allows us to be happy, very important in this day and age, and to keep our brains healthy. Okay, so exercising and diet is really important to keep your bacteria in your gut healthy. Okay. In E. coli, many, many years ago, back in the 1980s, technology had allowed relatively inexpensive DNA sequencing. Up until that point, DNA sequencing was quite expensive, so we could only sequence small regions of DNA. But with the advent in technology, scientists were now able to sequence lots of species, not just animals, but bacteria, other prokaryotes, and archaea, for example, in plants as well. Now, scientists are a bit nerdy, they can be, and so when they get DNA sequenced, they will sit and sort of peruse it for hours. These days, they might use a computer. But back when I was doing this stuff in the early 1980s, we would sit and look at these very interesting protein and DNA patterns that would show up on films. And we would count out the letters, and then we would look at the sequence of the letters to look for patterns. And so what the scientists identified back in the 1980s was that in the genome of E. coli, they discovered an interesting part that had these little, what are depicted here as black boxes. And what these represent are palindromic repeats. Okay, so radar, the word, for example, is a palindrome. It reads the same backwards as it does forward. Okay? So these palindromic repeats in DNA, if they are turned into an RNA molecule, the RNA has the ability to form what we call a stem loop, the secondary structure. And scientists, when they noticed that, were like, this is really unusual. Here are these stem loop type repetitive structures, and they're evenly spaced out. And we don't know why that is, but because they found it in this species and many other species of bacteria, they reasoned, well, you know, if it shows up again and again, evolutionarily, there's probably a reason for it. It's probably important. Okay? Fast forward about 10 years, and as the genomic databases, the information on DNA sequences from many different organisms grew, and as the power of computers grew, scientists were then able to ask the computers to do searches. And what they discovered was that these little bits of DNA sequence in between these repeats weren't really part of the bacterial genome. They belonged to things such as bacteriophages, which are viruses, and they seem to differ between different strains of E. coli. So the scientists put two and two together and came up with what they would say is a hypothesis or a prediction. And they said, I wonder if these sequences that come from bacteriophages, we'll talk about those in a minute, whether they are being remembered by the E. coli, and this is some sort of very primitive immune system, so that these bacteria can then be protected from subsequent infection by a virus, okay? Kind of like we would be. Okay, so this actually bore out, and in 2007 it was shown to be true that this structure was very important to allow an adaptive immune response in a bacterium. Okay, so the name CRISPR, you'll see how this technology in the next slide is used to perform CRISPR engineering in other species. But this technology comes from the acronym here, clustered, because these are all clustered together, regularly interspaced, because these are all neatly spaced out every 30 bases, 30 to 50 bases or so. Short palindromic repeats. So that's the little black boxes here. That's where the name CRISPR comes from. Okay, so how does CRISPR work? So shout out here to the library. This is, show of hands, who's ever read Aerosmith? Nobody? Oh, good, a few. Oh, fabulous. One of my favorite books when I was younger. Okay, this was actually interesting to me because the hero of this book, Dr. Aerosmith, was hell-bent on using bacteriophages to try and treat an illness that broke out at a Caribbean island. It's a very good book. It won the Pulitzer Prize in 1926. I hasten to add I wasn't born at that time, but maybe not far afterwards. The bacteriophage in question are shown here. So this is a scanning electron micrograph, basically a photo. These big things here are E. coli. And these little dots that are sort of stuck on the surface of the E. coli, they're viruses. They're called bacterial because they attach to bacteria. And phages from the Greek meaning to eat. Okay? And then normally when these bacteriophages, what they will do is attach to the wall of the bacterium. And as shown in this cartoon, they will inject their viral DNA into the bacterium. In many cases, that will replicate and then make more virus. The bacterium might burst open and release more little viral particles. They look like little lunar landers that can then infect other bacteria. But unbeknownst to many people until the turn of this century, it turns out bacteria have an immune system to fight these guys. So what they do when a virus infects a cell is its DNA is actually recognized by the cell, and it gets cut up into some very special small pieces that are inserted into this thing called the CRISPR array. So this is this region, once again, that has these palindromic sequences, and then these little colored parts here are derived from different viruses and some other things that will infect bacteria. The bacteria then use this sequence, they make RNA from it. This RNA then with these little stem loops is processed, and you get these little guys down at the bottom here. They're made of RNA, and these colored parts are derived from the sequence of the invading viruses. When these little stem loops associate with proteins, and there's only one protein that's really important in this case, it's a protein called Cas9, then that stem loop and the sequence that recognizes the viral DNA can be brought together, and CRISPR will then cut up the viral DNA. And by doing that, it destroys the ability of the virus to replicate inside that E. coli cell. So this is a very remarkably sophisticated and elegant way that prokaryotes, that is bacteria, have evolved to protect themselves against infection by viruses. Okay? Any questions about that? Okay, so CRISPR, once again, then, it's an acronym that has been repurposed by the scientific community. Although CRISPR really stands for this region where the foreign DNA is integrated, we've repurposed it to refer to the ability of this molecule to cut DNA. And instead of cutting viral DNA, we're going to use it to cut, say, for example, human DNA or all sorts of other types of DNA. So that's illustrated here. On the right is, let's say this is a bit of double-stranded DNA shown in blue. And here you can see the helix shown underneath, which in this case has been cut. Both strands have been cut. What the Cas9 protein from the bacteria does is to bind to the DNA, and it binds to the DNA depending upon this little stem loop structure and this part here, which is called the target sequence. So the homology or the interaction between the RNA here and this DNA sequence is what directs this protein that has these two pairs of scissors here, molecular scissors, to this part of the DNA. And at that location, it will cut both strands of DNA and cut it into two parts. So imagine this DNA would extend a long way here and extend a long way here. The key thing is that by programming the sequence of the RNA here, we can tell it where to cut within a genome. So we can target this Cas9 cutting activity pretty much, with a few exceptions, to specific locations within the 3,000 million bases that we have in our genomes. It's truly a remarkable thing. If you think about it, if you can take together the protein and a little bit of this RNA called the single guide RNA, and you can introduce it into cells, either in an animal or in culture, and then this small machine will scan 3,000 million bases and find the right match, and then make a double-stranded cut. It really is remarkable. Nature never fails to blow me away with stuff that when you step back and think about it, it's like, wow. Okay? All right, so all we really need then to be able to program where we want to cut a piece of DNA is the knowledge of the sequence of the genome of the organism, say the mouse or a human or a plant or a tomato. Okay? The availability of a suitable target site, and those again are pretty common. We need to have some of this Cas9 protein. You can actually buy it commercially. And you need to get some of this single guide RNA. Again, you can buy this commercially. If you go online, you can look at a company called IDT DNA. They're based in Coralville in Iowa. They're very careful about who they sell to, okay? For reasons that we'll talk about at the end. But this has made the genetic engineer's life much easier for being able to engineer changes in genomes. Okay, any questions about that? Okay? Yes, sir.
It seems to me that you might get a little bit of interference if you have a gene that, say, you're trying to repair as a pseudo gene.
Yes, so what's your name? What's your first name? Jeff. So Jeff is very knowledgeable here, and Jeff is quite right. In our genomes, we often have copies of genes that are very similar, but maybe not identical. So Jeff's question is related to how can you be sure that you're going to hit or target one gene and not a closely related gene? Algorithms come to our rescue. Companies such as the one I mentioned, plus various other institutions around the world, through trial and error, have been able to allow us to compare the sequence that we propose to cut and to look for what are called off-target sites. And so we can minimize the likelihood of that happening. Nonetheless, what we usually do, and we do this in my own lab, is that we will go and look at the top 5 or 6 off-target sites after we've done an experiment, and we will sequence them to make sure there has been no CRISPR activity there, and the CRISPR activity is only where we want it to go. But yeah, great question, Jeff. Okay, so some examples of the use of CRISPR. When you take this Cas9 protein and this guide RNA and you make a double-stranded break, normally what will happen is the cell will try and repair that double-stranded DNA break immediately. DNA double-stranded breaks are bad news for a cell. It sets off alarm bells. The cell doesn't want to have chromosomes being recombined. It may be precancerous, so it wants to repair these quickly. So what they will often do is some proteins will bind to the ends where the cut has occurred, and they will stick them back together as best as they can. Sometimes that will remake the original sequence, but if that happens, it will usually just get cut again. So what often turns out is that you get very small changes in the DNA sequence here, maybe insertion of 1 or 2 bases, or removal of 1 or 2 bases. And so we have a word we call indel, standing for insertions and deletions, that may occur at that site. This is a very efficient way of destroying the function of one particular gene. And I'll show you examples where we can so-called knock out a gene's function using CRISPR-Cas9 without having any repair performed other than sticking the ends back together. The other example shown on the right here is what can happen in our bodies and in bodies of organisms where they have two chromosomes that are the same. Yeah, you get one from mom, chromosome 1, say, you get one chromosome 1 from dad. If you cut one copy of a chromosome, then the other chromosome can act as a repair template. So a wacky analogy, let's say my favorite James Beard cookbook, somebody tore half a page out of it halfway through, and I'm like, oh no, I'm not going to be able to make that recipe. You know, how could I fix that? Well, I might go and find a friend's copy of the same book, and then I may Xerox that page, and then go stick that page back into my book as a way to repair it. Okay, so a wacky analogy, but hopefully you get the idea then that when you cut one chromosome, the other chromosome can also act to repair the one that's been damaged. And scientists have used this effect, and they can introduce extra DNA into the cells such that we can cause a change that we want to have occur in the DNA rather than having the original DNA being reformed. Okay, so these with good old-fashioned Cas9, it's only 10 years old, but the standard Cas9 and standard cutting, you either get little indels that usually destroy a gene's function, or with the assistance of some extra DNA added, you can introduce changes that you want to have in that genetic DNA. Okay? All right, so here's one example. This was on the first slide. There's a company in Dallas. It's called Colossal Biosciences. It's started by an entrepreneur in Dallas as well as somebody at Harvard, George Church, who's a very famous scientist. What Colossal wants to do, their goal is to try and de-extinct species that are extinct. So they want to bring back, for example, the woolly mammoth, or the thylacine, that was the Tasmanian tiger, the dodo, a variety of other species. Okay, and they might think, well, that's a bit crazy. You know, they're probably extinct for a reason, but there are other reasons that you may want to do this. So African elephants, for example, are under significant stress. And so the ability to try and keep species alive and to change their DNA to allow for fitter organisms is something that's very important. So what these scientists did, they used CRISPR to change the sequence of 7 different genes, and they did this all at the same time. So it shows you just how multiplexed or how incredible this can be in making up to, in this case, 7 changes in the mouse's genome at the same time. These changes affect the color and the waviness of the hair. So here on the left then are sequences in 7 genes that have been changed to mimic those found in woolly mammoths. This animal doesn't have tusks, and you know it's not going to invade Siberia, but the point here is to show proof of principle, and these scientists are actually trying to use this now with elephant stem cells. So they are trying. It'll be a great party piece. I'm a little bit wary of it, but if they can one day produce an elephant that has a very long, shaggy, hairy coat, maybe I'll find a place with climate change that'll be quite happy. Okay, so an example of gene editing in mice. So the existing applications of CRISPR genome editing, then you can break them down into 3 categories. There are what we call preclinical or basic research. There are biotechnology type applications, and then there are clinical therapies. For the preclinical research, this involves work that I'm actually doing at UCI with a lot of very talented people that I get to work with. We use the mouse as a model organism to study human biology and human disease. We're very interested in Alzheimer's disease and trying to develop treatments ultimately to help combat that. It's an extremely debilitating disease, along with many other neurodegenerative diseases. And so we want to try and develop effective therapies. Well, you can't develop the therapies until you have a good animal system to try and evaluate things on. So what we've been doing over the past 10 years now, almost 10 years, is to change certain genes in the mouse to give them the human versions of the genes. And these are genes that are involved notoriously with development of Alzheimer's, such as your ApoE and your tau gene and your amyloid genes. And we haven't published this yet, but I'm happy to say that we have excitingly some new strains of mice now that are developing amyloid plaques in the part of the brain that you would normally expect them to happen in humans at the age in the mice at which they would be happening the respective age in humans. So we're optimistic that that advance, if it continues, fingers crossed the NIH keeps going, will allow us to make inroads into generating a model which can then be used by the drug companies for gratis. This Model ED program is sponsored by the National Institutes of Aging. We get about $6 million a year at UCI to develop these animal models, and we're charged with making all of the information and all of the models freely available throughout the world to pharmaceutical companies as well as basic researchers. So the idea here is that we want to accelerate the rate of discovery so we can bring new treatments to the marketplace. Okay, so biotechnology, you can edit crops and livestock to improve fitness, disease resistance, and to improve nutritional value. I'll give you some examples of those. I'm not going to talk about this, but there have been CRISPR used in pigs to remove proteins on the surface of cells so that organs can be transplanted in humans for short-term purposes, people needing organ transplants for liver or kidneys, etc. And then, again, work first pioneered at UCI by one of my colleagues, Tony James, and this is development of something called gene drive to try and suppress transmission of malaria. And this is something that's slowly coming our way. And you may have noticed Aedes aegypti mosquitoes nearby, so it's only a matter of time until those sort of diseases, sadly, are in this part of the world and not just over in Florida. And then the last part here I'll touch on towards the end on our genetic modification or gene therapy in humans. Okay, so I mentioned at the start about using CRISPR to lower the glycemic index of rice. Rice is high in starch. What you can simply do here is to use CRISPR to inactivate or deactivate a gene that makes starch. You then end up with rice that has a higher amylose content, but its glycemic index, or the amount of sugar it generates when you eat it, is significantly lower. So for the over 500 million adults worldwide that suffer from diabetes, this is something, another option. You don't just have to eat brown rice, or you don't have to eat other things with high fiber. Many people in Asia, for example, are dependent upon rice as a staple. So one of the benefits already then of CRISPR in agricultural uses. Okay, another one, tomatoes, yum yum. Okay, hands up, who's been really saddened when they look at a beautiful tomato, or tomato to you, potato, and you take it home and then you bite into it and nah, yeah, it tastes like water, yeah. So over many years, tomatoes have been selected to be bigger, but the trade-off there is we lose a lot of flavor. So some very clever scientists in China have looked at the tomato genome, and they use CRISPR to deactivate 2 genes that now lead to the tomatoes having a much higher sugar content, being much sweeter, and they also retain more flavor. Okay, so it's an interesting point of discussion perhaps at the end, how many people might consider these as genetically modified organisms or GMOs. Yeah, and so in Europe right now, things that are CRISPR really aren't considered GMO. Our government may be a little bit different. It'd be interested to hear your opinions. Bananas, more popular than oranges and apples combined, yeah, in terms of consumption. There is a fungus that is moving slowly from Africa over to the Americas, and this fungus decimates banana crops. And so this is started to show up now in Colombia and South America, where bananas are particularly important. So CRISPRing has been used with banana plants to engineer a change in the genome to make them resistant to fungus. Okay, so another significant advance. The last one here on the right, this actually comes from research that started in the mouse many years ago. Scientists mutated a gene called the myostatin. They actually named it myostatin afterwards because when they knocked out the gene in the mouse and bred the mice, they found the mice developed and looked like Arnold Schwarzenegger in his prime. They had massive amounts of muscle. Agricultural people have known for many years, the Belgian blue cattle, for example, have extremely large muscles. And when they looked at the genome of the Belgian blue cattle, lo and behold, the same gene had been affected. So this is now a party piece that many scientists in the food industry are disrupting this same gene, myostatin, in different species. It's been done in sheep, it's been done in other strains of cattle, it's been done in rabbits, it's been done in mice, and now here it is in the Japanese market, is done in sea bream. So the Japanese market really loves sea bream. On the right, at the same scale, here is a non-modified fish. And on the left, you can see, if you look on the side or on top or tipped over, that this animal has significantly more muscle mass. And something that was fascinating to me is that the consumption of food by the non-modified and modified animals is the same. So for the same amount of feed, you have additional protein then that you can use. So this may be one way of trying to treat the big problem we have of too many people on too small a piece of real estate. Okay, so what about human disease? So what I'd like to do in the last 10 minutes or so here then is to introduce you to a now famous woman. Her name is Victoria Gray. She's African American and she suffered from sickle cell disease. Okay, she is now cured. And she is a poster child and goes around giving talks. Sickle cell is a nasty disorder. It tends to show up in the African American population. Roughly 1 in 400 African Americans has sickle cell disease. What happens in sickle cell? You have a mutation that affects one amino acid. And that causes this hemoglobin protein to change its shape very slightly. And what that causes in turn is that these nice red blood cells that should look like this, they end up turning into little sickles here. And then these sickled red blood cells, unlike the normal erythrocytes or these red blood cells, they can't get through the vasculature very easily. They get clogged. So what happens when they get clogged is that you get infections, you get really bad pain crises, and you have loss of energy and fatigue. These are extremely debilitating symptoms that require treatment in hospital. Individuals will often have at least 5 so-called crises a year, and it's not only debilitating, it costs a huge amount of money to treat. Okay? So there is, the idea then was, can we use CRISPR to somehow treat this disease? So I need to take a step back for a second and explain a little bit about what happened to all of us as we were developing inside our mom's wombs, and then what happened after we were born. So during fetal development, because the amount of oxygen you have inside your mom's womb is a little bit different from the outside air, you make a special type of hemoglobin. This is a protein that carries oxygen in red blood cells around your body. This is called fetal hemoglobin. And here, prior to birth, so 6 months, 3 months at birth, you can see the fetal hemoglobin normally goes down, and then the adult hemoglobin takes over, and it is produced and rises around the time of birth and takes over from the fetal hemoglobin. Okay? The problem with people that have sickle cell disease is their adult hemoglobin doesn't work properly. To make a long story short, scientists, by studying many people with this genetic condition, showed that some people had really bad pain, and infections, while others weren't so much affected. And it turned out that the ones that weren't so much affected, their bodies were still producing the fetal hemoglobin after they were born. So scientists then figured out, well, how can we then continue to cause the fetal hemoglobin to be produced in individuals that have sickle cell disease? And could this be a cure for this disorder? So this is a little bit of detail, okay, but don't freak out, stay with me. Here's our fetal hemoglobin. It's made up of these two alpha chains or subunits and these two gamma subunits. The adult hemoglobin has the same alphas, and you just turn on beta globin instead of the fetal globin. Okay, and here's that graph you saw on the last page. Here's the fetal hemoglobin normally coming down, and here's the beta globin coming up, but this is the one that gets affected in sickle cell conditions. And so normally around 3 months after birth is where symptoms of sickle cell disease will show up. Turns out that the fetal hemoglobin production is turned off by this protein. Okay? It's what we call a repressor. And it says, stop making fetal hemoglobin and start making the beta globin instead. So scientists went, okay, well, how about if we turn off this protein and maybe you'll still make fetal hemoglobin? It works. Okay? So when you use CRISPR and you destroy the activity of this protein and make sure it isn't turned on in the red blood cells, those red blood cells continue to make the fetal hemoglobin, and those red blood cells now don't have that abnormal shape. Okay? Here's how it works in the clinic. Individuals with sickle cell disease will show up in the clinic, and if they're approved for these treatments, the scientists and the clinicians will take a bone marrow sample from them, and in that bone marrow there are stem cells. And those stem cells, if you put them back into a person, can turn into all the blood cell types in your body. What the scientists do then is to take those stem cells and they treat them in the clinic with CRISPR, and they knock out or inactivate that part of that gene for this protein called BCL11A. And then they put those cells back into a person after they've removed the marrow. They get rid of the endogenous marrow, but they put the person's stem cells that they took from them and replace them back in the same person. Okay, so there's no need for a bone marrow transplant from unrelated individuals here. You're using your own stem cells and putting them back in. So that prevents any issue to do with rejection. Okay? When you do that, I've simplified the data here. So this is from one patient. What you're looking at the top here, these different colors are the different types of hemoglobin. The S is a sickle cell hemoglobin. So this patient, prior to being treated, she was making roughly 50% sickle hemoglobin and not much else. Okay, this normal adult hemoglobin was almost completely gone. 15 months after infusion or replacement of those cells that have been treated by CRISPR, look at what's happened. You still have the sickle hemoglobin being produced, quite a lot of it, but now you also have the fetal hemoglobin being made. And so the combination of these two, even though there's sickling hemoglobin around, this fetal hemoglobin can overcome it. And so the red blood cells have a normal shape, the pain crises go away, no more infections, and these people can lead a productive and happy life. Okay? So this was approved by the FDA a year and a half or so ago now. To date, roughly 31 people have been treated with sickle cell disease. 29 of these, it has been successful. The other individuals are still alive. Just the treatment wasn't successful, but they may have to be treated again. Okay, so these patients have been free of symptoms without any significant side effects since transfusion with these CRISPR-modified cells. Okay, so this was one of the first examples. There was one slightly earlier to do with a liver dysfunction that's equally debilitating. but I thought this is a good example to give you because many of you may be more familiar with what sickle cell disease is now. All right, so in the last part here then, I'll tell you that most of the genetic diseases in humans are usually caused by very small changes in the DNA. So if you look at this pie chart, just look at the blue and the red and the green here, that's roughly 75%, and these are single base changes or changes involving 2 or 3 or 4 bases. They're really small, okay? So, for example, here's cystic fibrosis. I know some of you may be familiar with this. It's commonly associated with removal of 3 bases in a big gene, and that is quite common in the human population. And so cystic fibrosis is maybe 1 in 2,500 live births. These individuals then have a buildup of mucus. They suffer from infections. It'd be great if we could try and treat this to cure the condition instead of having to treat with antibiotics and massages and intubators. There's another one down here that's caused by addition of 4 bases. This is called Tay-Sachs disease. It's common in the Ashkenazi Jewish population and in certain populations in Canada and also in the southern US. It's a devastating disorder that can affect young infants. They usually don't live much past 5 years old in the most severe cases. But again, if we can then get the CRISPR into the cell, perhaps we can try and edit these changes and reverse them, thereby curing the individuals. So this is the goal. Now, up to now I've told you that CRISPR will generate a double-stranded DNA break, and that's been great for people with sickle cell disease. That editing of the gene may differ a little bit amongst people, but it always, for the most part, works. But, as I've just shown you in the prior slide, there are cases where we want to be very precise about the changes. For example, in cystic fibrosis, we just want to add those 3 bases. In Tay-Sachs, we just want to remove these 4 bases. And in sickle cell, we just want to change 1 base. The problem with standard CRISPR is that when you generate a double-stranded break in DNA, it sets off alarm bells inside the cell. Okay? There are all these sort of protein signaling systems that happen, but basically the cell goes, we're in problems here, we're in damage mode. And that may not be good in the long run for therapies. It's okay in the laboratory when we're engineering mice, because we can tolerate those changes and we can select for mice that have what we want. But it's no good in a clinic where you don't want to do any harm. So scientists then figured out another way to use CRISPR that instead of making a double-stranded DNA break, you just make a single-stranded break. Okay, so here's a standard CRISPR, as I showed you earlier. Here, once again, is our protein. And here's an RNA directing cutting of the genomic DNA, shown in blue, at 2 sites. So what the scientists did was to change the sequence of the protein shown here, so now it doesn't cut here anymore, it only cuts in one place. So instead of generating a double-stranded DNA break, you now have what we call a nick. And this nick in the double-stranded DNA backbone allows this part of the DNA to be freed for a period of time. Okay? What it doesn't do is set off the alarm bells inside the cell. Scientists at Harvard, and in particular Dr. David Liu, I would encourage you to, if you're interested, look him up on the web. Has started a company called Prime Editing. I anticipate that this, what you're looking at now, is going to be the way going forward for CRISPR gene therapy for the next 5 years or so. How does this work, and how is it different from standard CRISPR? The 2 things I want you to notice here is that this guide RNA that helps to direct the Cas9 to the DNA, it's a little bit longer. Okay, we call it a PEG RNA because it has a primer editing guide. In other words, this little tail here can bind to the part that is being released of the DNA and can allow synthesis of new DNA depending upon the sequence that we introduce here. So when you use Cas9 that has this particular enzyme attached to it, it's called reverse transcriptase. Basically all it does is to add DNA upon an RNA template. You can add new nucleotides down here, such as shown in red, the sequence of which is dependent upon what you include in this RNA template. Okay? So the example shown here then is where you can add 3 bases here using these so-called prime editors. And it turns out that in work that's currently underway, you can add up to 20 to 30 bases, or you can remove 20 to 30 bases as well. And remember, 75% or so of all the mutations in humans are 1 base change or maybe a small deletion. So that's why this sort of technology now, I think you'll find more and more in the literature and in New York Times and on news, you're going to find that this is what's going to be used in clinics to treat a whole bunch of different types of disease. Okay? So the last part I'll show you here then is proof of principle. You remember our red blood cells and the sickle cell disease? This is just the percentage of edits inside a population of cells in a dish. So this hasn't been published yet. It's been worked on in animals like mice and other species, but right now it's been done for proof of principle on cells cultured in a dish. If you take the hemoglobin variant, this 1 letter change, you can, with this prime editing, generate about 50% recovery of the correct base sequence. Okay? Over here with Tay-Sachs disease, this extremely devastating, debilitating condition that causes infant lethality, you can now get anywhere, depending upon the experiment, between 20 to 40%. And the scientists are not content with these. They're continuing to push the system to make it more effective and, to Jeff's point earlier on, to make sure there are no off-target effects. Okay? So just 2 examples. There are many more that Dr. Liu's lab has been working on, and I suspect these are now already making their way into preclinical trials. All right, so the last slide here is really considerations about the use of CRISPR. So on the left here are what's happened in the past 10 years or so, roughly now, of CRISPR. The first sort of CRISPR knockouts in animal species were really done about 2013, 2014. So we're about 10 years in. We can do knockouts of genes. We could do sickle cell therapy. We can modify genes in mice, in crops. We can do a variety of other things with them, including more than 1 gene being modified at a time. That's now in the past. In the next 10 years, here's a list of things that we would like to see. This has already been accomplished. Approval of sickle cell therapy by the FDA, as well as some other disorders. This now then is in process, and many other diseases have been accomplished. Nutritional value, I showed you some examples of that. So this is already on the way. This, to me, is the big bottleneck. You can do this in cells in a dish, but getting it into the organ that's affected in humans that have different diseases, that's the big challenge now. But I'm confident that scientists, with ingenuity and with a lot of hard work, there's famous scientists that say, I'm amazed, the harder I work, the smarter I seem to get. Yeah? Same thing here. If we apply ourselves, we can improve technology for in vivo living delivery that will allow this to become a reality in many species. Multigenic traits, you saw the woolly mouse, and you can see the big fish. Crops and disease resistance, all of this is now. This came out maybe 4 years ago. All of that's already been done. So this technology is advancing at a rapid pace. So the last thing here on the right, I've listed a series of things that you might want to think about in going forward. To Jeff's point earlier, unintended consequences, off-target effects of these things. We try as hard as we can, but we can never fully exclude the likelihood of having unintended consequences. With any sort of medical treatment, there's a risk to benefit ratio. Okay? And you always have to weigh that in mind and make the patients aware of it. But the key thing here is we don't fully know all the time. We can't be certain. We try our best. Long-term effects, we really don't know. The only way we're going to find out is by watching, tracking people carefully and seeing what are the sort of changes that occur. Ethical concerns. Okay, hands up who's heard of designer babies. There you go, many of you, yeah? So, in places that are maybe somewhat unethical, or you may have people coming in saying, oh, we can screen your fertilized eggs, and we'll make sure, and we can even edit them to make sure your kids will have blue eyes or this, etc. This has already been done in China, in one famous example a few years ago. It was harshly received. It may be going on in places, but personally I would not advocate for that. Socioeconomic disparities. I'm sure many of you are only too painfully aware of the cost of healthcare. Don't get me started, it's run by insurance companies and middle people, etc. But think about the African American community in particular, many of whom are in the lower socioeconomic status. One of these CRISPR therapies to cure stem cell, a sickle cell disorder, is currently listed at about 2.2 million. Okay? Why do the companies set them at that rate? Well, their argument is that over the lifetime of treatment, this is going to be cheaper than having you treated for your entire life. So to her credit, Jennifer Doudna, one of the co-inventors or applicators of CRISPR, is now trying to start a small non-profit to treat patients using this, and her estimate on the cost is about $220,000, roughly 1/10 of the cost. So, you can do the math as to where the profits are going here. Environmental impact, okay? You can't just go CRISPRing things and letting them run loose, be they plants or animals. This is something really important. There are too many lessons that we've learned over the years, bringing kudzu, for example, into the southeast. That was a train wreck. Taking rabbits to Australia, that was another train wreck, okay? So we're always trying to be cognizant. For example, the malaria treatments to try and reduce transmission of the infectious agent that causes malaria. We're not going to kill, scientists don't want to kill the mosquitoes because they're valuable for bats and a variety of other birds. But they want to immunize the malaria, immunize the mosquitoes so they don't catch malaria. Okay, but one of my colleagues at UCI, Tony James, he's currently trying to get permission to do a trial on the west coast of Africa to release these in a controlled study on an island to see what the effect might be. So this then leads into regulatory challenges. Okay, there's a lot of problems when the government see these things that are being done in the scientific domain, and they are understandably wary about what the risks may be of them being introduced. I mentioned, for example, you can buy Cas9 and you can order up RNAs from companies, but most of those companies are pretty savvy. They want to make sure you're at a research institution and you're not some person sitting in a shed somewhere that's going to do something nefarious with it. But regulatory challenges for a lot of these things are still in flux. Okay? And then the last one here, the security risk, as I've alluded to already, it's not out of the realm of possibility that bad actors could leverage these things and try and generate organisms that are particularly pathogenic. That's a real worry, but again, we try and do the best we can. As technology advances, it comes with it rewards and it comes with it risks. So I'll leave you then with this sort of take home. There's a key ongoing objective is to try and balance these benefits and risks to harness the potential for the greater good while mitigating or minimizing the potential harm. Okay, so thanks very much for your attention. Sorry, I ran over a little. If there are any other questions, I'll be more than happy to try.
Yeah, and I think we have some microphone management going up and down. So raise your hands. Grant, you can pick them out, and we'll have the delivery by Kunga and Jerry.
Hi. So thanks for a really fascinating presentation.
You're welcome.
You mentioned that you're getting some funding. I'm just curious how much of your funding comes from the government and what you're anticipating in the current political environment.
The short answer is...
That's a non-science question.
Yeah, I'll keep it brief. The short answer is we don't know. We really don't know. I'm most concerned about the appropriations and for the upcoming budget. And I'm truly concerned that the NIH is going to take a massive hit, the National Institutes of Health. In brief, I would inform you, if you don't already know, economically, for every dollar that the NIH puts out, the US economy generates at least $2.50. So there's a 2.5 to 1 return on investment. I hope that level heads will prevail and that people will recognize that there is an awful lot of really good work going on that drives, it really is an engine that drives the US economy.
Great.
Is there any application of CRISPR technique for cancer?
Yes, that's a great question, and that is an approved therapy already. So one of the nasty things about cancer is not one disease, it's hundreds of diseases. One of the common things with tumors is that they like to suppress the activity of the T cells. So the T cells are part of our white blood cells that go in and attack things and get rid of things that the body doesn't want. When T cells trying to invade a tumor environment, the tumor can often send signals to the T cell saying, hey, calm down, or nothing to see here. Okay? It happens through a particular type of signaling pathway, and scientists and clinicians now can use CRISPR to inactivate the hearing of the T cell, if you like. So when they take T cells from a person, they can turn off the T cell ability to listen to the tumor. And when you put those T cells back into a person, they can now attack the tumor. That's just one example of immunotherapy for cancer. That's a lecture in itself. I would encourage you to try and invite one of the Cancer Center members. We have a National Cancer Institute accredited academic center. It's the only one in Orange County. Okay? City of Hope is great, but they're not an academic institution. We have an NCI accredited center where there are basic researchers working side by side with clinicians. And that is a recipe for creativity and success. So I would encourage you to consider perhaps inviting one of them to come out and give you a talk on advances in cancer diagnosis and cancer treatment. Yeah, I think you'd be truly amazed at the advances that are being made. Yes?
Can you give me some insight of what's going on a molecular level when that cutting happens?
Yeah, well, yes. So DNA, let's go all the way back to the start very, very quickly. DNA is a double-stranded helix, and it's made up of a phosphate backbone, and it has a sugar here. This is called the ribose, or deoxyribose in the case of DNA, and then these bases. So what happens with CRISPR is that the CRISPR, first of all, will recognize the region that it wants to bind to through this RNA here, and the enzyme here, this protein called Cas9, causes the DNA to relax. And it causes the DNA sequence to unfold. It forms what we call an R loop. When that loop opens up here, that allows this guide RNA to come in and to bind to the reciprocal, the recognized sequence. At that point, these two small regions of the protein literally act like molecular scissors. And in the backbone of DNA, there is a chemical linkage. It's called a phosphodiester bond, and it cuts at that site. And by breaking that site, it then leads to scission of the DNA backbone. In this Cas9, there are two such sites, so it causes a double-stranded break. In the one I showed you later, the prime editors, the investigators have destroyed one of the enzyme activity, so you only now get one cut that gives you just a nick. Okay? So these proteins are really enzymes, the little molecular machines. They can recognize with remarkable accuracy a sequence, and they can cause a cut that breaks the DNA backbone. Does that help?
Recognized by the fields of the various
Yeah, it's
atoms lining up.
exactly, yes. So if we go back to the first slide, I mean, that's such a great question, because clearly what you're trying to get at here is how do bases recognize each other? So you can see that thymine and adenine, TNA, they have 2 hydrogen bonds, and guanine and cytosine, they have 3 hydrogen bonds. And then you see the sequence here is somewhat unique. And so literally these 2 sequences here, if you put them together, they would sort of shuffle over each other thermodynamically and look for a condition where they were happiest. And that thermodynamically, for the people in the audience that are familiar with this, it just minimizes the amount of free energy. So they're at an equilibrium and they can interact with each other. DNA, if you melt these strands apart, they will come back and find each other very quickly. Yeah? So in CRISPR, the bacteria have devised a way to utilize that system so that when you have an RNA here, it can also recognize the respective or the corresponding position in the DNA. And then you get that little cleavage, and that makes your break. It's, you know, I often fantasize about trying to be very, 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 very small and sitting inside a cell, watching everything that's going on. And there was this wacky movie back in the 60s,
Yes.
Fantastic
Voyage.
Yeah, you might
With
remember
Raquel
it.
Welch.
Yes, yeah. So, yeah, that's one of my fantasies. How not Raquel Welch, but the premise of being able to shrink yourself to a molecular scale where you could actually watch. As Yogi Berra used to say, you can observe a lot by looking. Yeah? So if you could actually watch what was going on inside a cell, just how much we would expand our knowledge of how biology works. Okay? That is actually going on at UCI as well. Not shrinking me, but using specialized microscopes to look inside living cells to understand how they work. Yes? Hi.
The illness of Parkinson's, the
Yes.
disease. Any updates on that you can share with us?
Parkinson's can be caused by a variety of different genetic causes. Classically, it presents when you lose the ability to synthesize a particular type of an amino acid-like protein, L-DOPA, in part of the brain. And that can lead to a tremor. There are some cases of Parkinson's that are due to inheritance of a dominant mutation. Those may be ones, there are very, very small numbers of those that could be treated. In terms of actually trying to treat Parkinson's, part of the problem is the part of the brain, the substantia nigra, that normally produces that L-DOPA, it atrophies. It basically dies off. So I'm not aware of any therapy involving CRISPR that could be used to rescue that loss of the substantia nigra, that tissue.
The deep brain stimulators.
Yes. Deep brain stimulation does. When I was at Emory University, there was a physician there, a neurologist, who was extremely well known for doing that. And what that does is to try and combat an imbalance in the nervous system that is due to the absence of L-DOPA. So yes, talk to your physician or neurologist, but deep brain stimulation, look it up on Google, it is often being used successfully to try and treat Parkinson's. Yeah, let's see, sir, I was one, come back.
Like sickle cell,
Yes.
type 2 insulin-resistant diabetes is heavily genetically predisposed.
Yep.
I wonder if CRISPR has anything to offer these people and where your research is in that regard.
That's a great question. I'm actually unaware of any developments in using CRISPR to treat type 2 diabetes. Type 1 diabetes, that's due to an immune reaction against the beta cells in the pancreas that make insulin, that's something that people are working on to try and knock out the particular genes that make antibodies that can recognize the insulin-secreting cells. And the idea is by blocking those, that immune response, you might be able to help ameliorate it. But part of the challenge in people that have insulin insufficiency with type 1 is that once the beta cells are gone, it's very difficult to get them back. That's a different lecture on stem cells. Okay? For type 2, I'm not aware of any CRISPR modifications that are specifically being used. Classically, the best way, unfortunately, to treat type 2 diabetes is diet and exercise. Okay? And the other way to treat it is Ozempic. Ozempic, and I will tell you, Ozempic is not only good for treating type 2 diabetes, there is now very good evidence that Ozempic is also good for brain health. So
I'm going to ask.
Don't quote
I'm
me
going to
on
ask
that,
for
but-
one more question. We do have coffee, tea, and cookies, and we have the ability to stick around and,
Yeah, I
and
think
talk.
the
But I'd like
gentleman in
to.
the front here. We'll do two more. Okay. Does CRISPR have instruction? Great question. The instructions for CRISPR come from the sequence of the RNA. So this is an RNA-directed nuclease. This enzyme will cut the DNA, one strand or both strands, but it does so in the best case situation depending upon the sequence of the RNA. So you can order an RNA and say, I want to cut here within the whatever genome, say the mouse genome. I can go online and look at the genome browser and say, I need to make an edit in this gene. And so I'll order up an RNA that has this sequence and then specifically has this, so I can do that ideally at that one location. And again, it's amazing that it does it with this accuracy and with speed. Yeah. Yes, sir.
Yeah, great talk.
Thank you.
My question is not embarrassing to me.
No. No question. No question is embarrassing.
Some years ago, Chinese scientists allegedly artificially fertilized, and maybe the wrong terminology, a human embryo.
Uh-huh.
And
It's a clone,
there was
right?
a huge outpouring of concern across the world
Yep.
that maybe we were unleashing a monster.
Yep.
Is there any validity to that?
So I think, I could be wrong, but I think the incident to which you're referring is where a set of twins, as pre-implantation embryos, they had been treated with CRISPR to inactivate a gene that is important for infection by human immunodeficiency virus, or HIV. And this Chinese scientist wanted to show proof of principle that you could knock out that gene and then generate infants and people, essentially, that are now resistant to HIV infection. Okay? It was successful in terms of knocking out the gene. I personally have not heard of any reports of an untoward outcome with these now probably in their early teens, I would guess. But as one of the things I mentioned here is we don't know what's going to happen. I don't know if the genomes of those individuals were thoroughly sequenced, to Jeff's point earlier, to see if there was any off-target effects or unintended consequences. So that was done without significant regulatory oversight and approval. It was thought to be extremely capricious and dangerous, very risky, and it's still held front and center by the community. So any sort of trials that are done in humans, as far as I'm aware, certainly in the Americas and in Europe, are done with very rigorous regulatory oversight.
Okay, I'd like to formally draw the session to a close. Thank you again, Grant. As I mentioned, there's a little post game going on back there, and don't forget, if you like this, come back on April 30th. We have Lucy Jones to talk about tectonics. See you then.